Everyone, my name is Mauricio Leon Mendez. I am the current chair of the Environment and Energy Research Council at the Center for Transportation Studies. My full-time job is at the Minnesota Pollution Control Agency as an economic policy analyst, and I'm glad to be here today to welcome you to today's webinar and meeting sponsored by, of course, our CTS, Environment and Energy Transportation Research Council. So a little background on the councils. The CTS councils provide a forum for transportation professionals and researchers to exchange information on current transportation issues and trends. They bring together university faculty and staff with practitioners from the public and private sectors to recommend direction and participate in improving the center's research, education, and engagement programs. Sam will share the link to the council's information in the chart shortly. Today's webinar features a presentation on species from feces, a new tool to identify bats in culverts and bridges. I will share more about this in a moment, but first I'd like to turn it over to our Council Vice Chair, Ross Pinter, who is co-facilitating this webinar. Ross, would you like to introduce yourself and cover some details on the webinar?
Hey everyone, my name is Ross Bittner. I'm Engineering Services Manager with the City of Edina, and I'm the Vice Chair for this council. So I'll give you all a little bit of housekeeping for how we're going to run the webinar today, and then preview some spring events for you. So at any time during this, we're looking forward to your presentation or your participation. You can put your Q&A in at the Q&A on the bottom of the screen. When we get to that Q&A time, you could also choose to raise your hand, and then we can unmute you so that you can share your question verbally. So this presentation is eligible for AICP certificate credits. If you need any of those professional credits, please feel free to check it out. We're going to upload it in the chat or send the moderators here a message. So some spring events that we want to get you ready for. We're holding 2 other transportation research webinars this spring that you want to take a look at. One is on EV infrastructure and fuel policy, understanding transportation and economic impacts. The other one's how infrastructure shapes driver behavior and pedestrian safety. So check those out at cts.umn.edu events and seminars. Got the link there on the screen. We also are hosting 2 in-person events. On May 5th, we're hosting the All Councils meeting, awards and luncheons. And those are at the UMN McNamara Alumni Center. You can register for those events on the UMN site. And then finally, if there's any students joining today, please send a chat to all panelists with your name. We like to track the amount of students that engage in the event. So thank you. Look forward to this presentation, and I'll be back as we get into the Q&A. But I'll turn it back to Maurizio to introduce our speaker.
Thank you, Ross. I would also like to share that Sam Han-Duville and James DeSota from CTS are also on today's webinar supporting the event. Thank you both. Bats frequently use bridges and culverts as roosting habitat, creating challenges for transportation agencies working to balance infrastructure needs with environmental compliance. Traditional visual surveys can be limited, especially when bats are hidden within structures or present only intermittently. This webinar will highlight ongoing research exploring the use of DNA analysis from bat feces, guano, as non-invasive method to identify bat species occupying culverts and bridges. Speaker Ron Moen will discuss how this approach works, share early findings from field applications, and explore how the results could support more efficient environmental review, project planning, and species protection efforts for transportation agencies. I am pleased to welcome today's speaker. Ron Moen is an associate professor in the Department of Biology at the University of Minnesota Duluth and a senior research associate with the Natural Resources Research Institute. His research focuses on mammalian ecology, including habitat use, movement patterns, and predator prey interactions, often using GPS radio telemetry. His current work includes studies on moose, Canada lynx, American marten, wood turtles, bats, and other wildlife, as well as projects related to climate change impacts and carnivore monitoring in the upper Midwest. Ron, now I turn the webinar over to you.
Okay, thank you. Let me share my screen here. Entire screen. Okay, yeah. Thanks for the introduction, and I'm happy to be here and present some information on species from feces. And the title came from Chris Smith with MnDOT, and I also call it Bats and Bridges once in a while. And kind of the motivation for this project was, like Mauricio said, when a species is listed under the Endangered Species Act and federal funds are involved, then the hope is that with what we learn, we can save MnDOT money in terms of any consultation with the Fish and Wildlife Service. So let's move on to kind of who we are. So this is me about 10 years ago, and Jessica Sieber is a geneticist on the project. Unfortunately, she passed away last October, which kind of slowed the project down a little bit, but we're back on track with genetics. And Raphael Stern is the statistician. He's much more than that, but that's what he is on this project, and he's down in the Twin Cities. And then I've got three graduate students who have worked on it. Jay Spartel is going to be doing some of the genetic aspects, and then Taylor and Mac did field work and a little bit. Their theses are actually on moose. In an army of undergraduates, and these students are the ones who are basically going out to the field and looking at bridges and deploying equipment. So what I kind of gave a little bit of the background, but basically I think that this project, because of what we're finding, is actually one of those projects that will save money. You know, it always costs money to do research, but it also costs money to manage and take care of the consultation with Fish and Wildlife Service. And I'll talk about a couple of the ways that we can probably do that throughout the presentation. And then I'll cover some methods that we use, and then look at bridge observations by MnDOT, because MnDOT actually did a lot of preliminary work looking for bats, and then we followed it up on this project. Add a little bit about what we're doing with acoustic detectors, and then finally get to the species from feces. So this is, I wouldn't really call it black gold, but you could think of it as that. It's what we're collecting from the bridges. First, I wanted to really talk about the background of why we're doing this project. In what essentially white nose syndrome has, as it says here, devastated K-bat populations. And it appeared in New York. It came over from Europe, where it has been present, in 2006. And within 6 years, it had spread to this blue line here. And the estimates are about 6 million dead bats in those 6 years. And since then, over the last 12 years, you can see the spread basically going all across the United States. One thing you could ask is, how can it spread so quickly? And the important point here is that it's cave bats. I'll talk a little bit more on the next slide, but basically cave bats hibernate in caves, which is a perfect place for white nose syndrome to be spread. So just like colds are spread among us. And how can it go so rapidly? Here's one example. The Milo is a little brown bat, which used to be the most common species we have in Minnesota. And it summers down here and actually migrates almost 400 miles up to here for hibernation. And you can imagine other bats doing that same thing, and it certainly doesn't take long when they make a trip like this every year to spread white nose syndrome. How devastating is it? 95 to 99% mortality after 3 years, essentially everywhere that the data has been collected. And that's based on acoustic work or examining caves. So that, and just for reference, COVID, and we know how that disrupted our lives, mortality rate for COVID was only 0.25% overall. So, I mean, this is literally devastating. And let's look at the species that it affects. So what I've got here are 7 Minnesota bat species. We've actually got one more that's appeared. Its range is moving north with climate change. But the bats on the left are all cave bats. And so these species are affected by white nose syndrome. And we'll start with the northern long-eared bat. It's called the northern long-eared because the ear tip, if you bend it, actually goes over its nose. And no other bat in Minnesota does that. So that's one way to identify it. This came from a project that we did about 10 years ago that I'll talk about. And these are all little bracelets to identify the bats and indicate that they have been handled at one point. And curious fact is that some of these little brown bats especially live to be over 30 years old based on these band returns. So that's a northern long-eared, listed as endangered in 2022, and it was first listed as threatened in 2015. So it's really the motivation for the project. But a second important aspect is that 2 other species are now proposed for listing. One species is a little brown bat, which, as I mentioned, used to be the most common bat, as you'll see based on the data I show. And then the tricolored bat, which has never been very common, at least in northern Minnesota. Finally, the big brown bat is about twice as big as these other species, and it is not as affected by white nose syndrome. Okay, so those are cave bats. These are usually called tree bats. So we have the red bat, the hoary bat, and the silver-haired bat. And these species are not affected by white nose because they migrate south and spend the winter in trees or in leaves on the ground. So they're not congregated where white nose can spread. So these are the characters that I'll be talking about in the data we're collecting. Again, I wanted to put some emphasis, some background information. So 10 years ago, white nose had just appeared in Minnesota, and we had a project with the DNR and with the Forest Service to look at maternity roosts. And the reason we looked at maternity roosts, that it was a focus, was that the listing emphasized these maternity roosts when the moms were giving birth and then lactating the pups, nursing the pups. And so the dates that the listing used was May 15 to August 15th as being most important, and that's when a lot of summer work construction work is done. And then they also emphasize tree clearing. So this would be if there's a maternity roost, for example, in a bridge, or even if a listed bat species is on a bridge, and tree clearing would be the same thing, because bats will also roost in trees. This is a transmitter that we used to find roosts. And the other thing we found in this project is that lactation based on the bats that we caught occurs from about June 15th to the end of July. And these are all northern long-eared bats with the state. And the young are nursing for about 4 weeks. Okay, what did we find? There's no bridges here. But what there is a lot of trees and a lot of dots. Each of these dots are roost trees. So the transmitters would last for 10 days to 2 weeks, and every day we'd go out and find where the bats ended up roosting. And these are all female northern long-eareds. So we'd capture them here. And then what they actually do is they use a collection of roost trees. This is probably from about 3 or 4 bats over 7 or 8 days. And so it's not just one roost that they use, but many. They have 1 pup per year, and we'd go out in the evening and watch the bats emerge. And the average size was 15, but a range of 1 to about 80 bats were in the roost tree. Another interesting thing, and that's why there's so many dots here, is that the females actually carry their young from one tree to another every other day, which was quite surprising. And the size is important here too. Essentially, they're foraging in about a half a square mile. It wouldn't always be square, but this is a good representation, and this is where they'd be roosting. So that's northern Minnesota. Here's southern Minnesota. I zoomed in a little bit because it's all agriculture on the outside here, but this is the same foraging area and the same cluster of roost trees. In the 700 meters, like basically that's the average distance to all of these trees. So what did we find about the northern long-eared? The most important thing probably is right down here at the bottom. I should have put it at the top. We had about 250 roost nights that we found, roost days rather, and there were no northern long-eared bat roosts in either buildings or bridges. In contrast to the little brown bats, and this is what's potentially important for the future, do roost in buildings and do I'm sure roost in bridges. But back to the northern long-eareds for a couple seconds. So these are examples of the trees. There's a little hole there. This is a bat. It just crawled out of that crevice, or exfoliating bark is what this one would be. And then here's a slit right down on almost ground level where some bats were roosting. Typically, they're larger trees, but they could go down to relatively small, and they used the common tree species. So down south, it was a lot of oaks. Up in the north, it was a lot of aspens, and other species too. So, but main thing is no roosting in buildings or bridges. So, but bats do use bridges. So what type of bridge do they use? The main reference for this is Keeley and Tuttle, 1999. There's been a few other reviews since then in papers, but they basically got it right with what they said. So day and night roosts, day roosts, they're spending all day, and those are probably the more important one. Night roosts are something where they're just landing on a wall or a ceiling and staying there and resting before going out to forage again. Characteristics, they're concrete. And that's because there's a rough surface that they can hold on to. Crevices had to be at least 12 inches deep, according to Keeley and Tuttle, and 3/4 to 1 inch wide. We're actually finding that they're using wider crevices. Have to be no water drips from above. And another thing that they suggested that was that they should be near or over water. One thing that's relevant is that the night roosts, because they're just temporary and resting, are probably going to be less restrictive. So, but we would go to the bridges and look up here for any evidence. And what we actually found most, and we used binoculars, so we'd see up top there and flashlights. What we actually found was that it was typically on the ends of bridges. And they'd be up in here. There's usually a crack. I don't know the engineering term for it. You can see one here as well, and that's where we would find the little pellets like this. And these were dropped from above. They actually look pretty much like mouse pellets. Rarely did we see bats. And what's likely happening here, probably out of the 300 bridges or so that we've been at, we saw them in half a dozen. And this was the only one that was really exposed. The others are hidden, like there's actually a bat here. You probably can't see it on the screen. Or they're even further in, and that they come out and they drop the pellets. If they're further in, we can't tell what species it is. The title talked about culverts, and we did an analysis of the MnDOT culvert database. In the definition, less than 10 feet of roadway travel distance. I had to download this from the MnDOT site this morning, but basically the travel distance would be here. And that would be less than 10 feet compared to a bridge over 10 feet. And many bridges are obviously longer than that. There's over 70,000 culverts in the culvert database, and this is the criteria from Keeley and Tuttle. And less than 100 of those 70,000 culverts are 5 to 10 feet tall, 300 feet long, and made of concrete. Metal culverts, the galvanized metal, just are not suitable. And MnDOT, and this is Chris and his team, have looked at some of these culverts, and they're only found in less than 0.1% of the culverts that they looked at. And so this is the kind of thing where the probability of finding bats in an area is one of the things that we'll be doing on this project too, and being able to extend it to bridges that we haven't seen. They found mostly big brown bats and then one little brown. So given this, we have not looked at culverts as part of this project. Really, they're not something that bats in Minnesota will use. Chris and his team have also done fecal pellet collection, and this shows a few things. They didn't see pellets in most of the bridges, and they found them, for example, in 91 out of about 1,600 bridges, over 6% in District 1 up here. The District 1 and District 6 were the ones with the most pellets found overall about 3% of the bridges. And one thing about the pellets, we don't know what bat species dropped them. That's what the genetics is for. We have some idea based on the density, and I was thinking as I was preparing for this talk, this is something I should look up, is how many fecal pellets does a bat deposit in a day, which would give us an idea of how much they're using a bridge as a place to rest, for example, because they'll usually drop fecal pellets as they're taken off. So that's something that was good for me to think about. Bridge types. So there's 87 different bridge types in Minnesota. at least in the MnDOT database. And 11 of those bridge types, and they're listed here, had 95% of the fecal pellets, and almost all of those were in this pre-stress or precast beam span type of bridge. And then 13 bridge types had the remaining 5%. So it's a limited number of bridge types that they're actually likely going to be in. Our collections as part of this project, we've collected in 2024 and 2025. We're also going to be collecting this summer. We've found pellets in about 50% of the bridges that we've looked at. So 240 not found, 226 found. And one of the important things about this figure is, to me at least, is a spatial distribution. These are the spatial distribution of bridges without pellets is essentially the same as the spatial distribution with pellets. And it's almost like you have to look at every single bridge from that perspective, which is another reason why this project could be beneficial. So then the second method that we're using is acoustic detectors. And this is an example of one hanging from a bridge. This is one of the easy installations because of these posts that are up on top of the bridge. We have cable so it can hang down below. And then this is a detector. We're using Wildlife Acoustics SM3 or SM4 models, and there's a microphone which picks up the sound. We lock the cables together, and then we put an information sign on the cable, which just indicates what the project is for. And surprisingly, to me, in one sense at least, we've had only one case of vandalism. We haven't lost any detectors, which is kind of what I was expecting going into this, especially when you get under some of these bridges and you look at see all the graffiti that's there. And it's not all the nice, well, relatively nice looking paint that you see here. So I expected things to be worse, but so far so good, knock on wood. So we deploy them for 14 days. That's what Fish and Wildlife Service requires for their inspection, or the things that they do. And here's another example of an installation. This detector is hanging here. In this case, you can kind of see here, we throw a rope over, and then we pull the detector up so it's out of reach, above the reach of a person, and we use a ladder to secure it, which probably helped in the sense of why we're not haven't seen vandalism. This is a sign, so it's clearly showing what it's for. What they're recording is kind of like if any of you use Merlin, the bird. They're essentially recording sounds that we can't hear. And this is an example of a little brown-backed call. And this is an example of a northern long-eared bat. This is time on the x-axis and frequency on the y-axis. Oops, hit the mouse and it does that. So you can see that there's differences, and we've got hundreds of thousands of calls recorded, and it's not something that you can look at manually. So we use this Kaleidoscope Pro software, which is approved by the Fish and Wildlife Service. And we also use Sonobat. So we use a couple of different versions of software. Not all calls can be detected either. I've got some data. I think I kept it in, otherwise I'll talk about it. In terms of, these are nice quality calls. They don't all come through like this because of how the bat's flying or background noise, etc. So what I'm going to show you next is a contrast of what happened with white nose, and also a contrast of forested bat detectors in the forest compared to detectors in bridges. So this is the forest, and these dots here are locations where we recorded bats right when white nose hit. It's actually 2015, 16, and 17. And then we're repeating the project this year to see the net effect. And then this is all the bridges where we've had detectors on, and essentially because the focus is northern long-eared bats, you can think of the forested area as stretching up kind of along where I'm moving the mouse here, and that's where we put put the detectors. Aren't that many bridges up in this area? Obviously, it's the boundary waters, so we can't sample bridges there. So let's see. Yeah, and this shows the years. So what we found, and this was, I double checked, actually triple checked and quadruple checked this because I found it so hard to believe. But looking at this top table here, here's an example of a detector in a forest setting. 2015, we were picking up 71 calls per night on average. In 2025, we still have the same number of calls. What actually astounded me was how many calls per night we were getting on bridges. And again, we just know that these are bats flying there. We don't know if they're staying in the bridges or they're just feeding, but it does give a indication of the species that are present. Looking at percent of calls per night. So this is the white nose effect. 10 years earlier when white nose first hit, and then now. And what's basically happened, big brown hasn't been affected. These are percent of calls per night. And we have a similar number of calls per night, so they're directly comparable. Red bat is never very common. The hoary, similar. Silver-haired really jumped up in relative proportions of the population. And one thing I will say is that differentiating between big brown and silver-haireds is not perfect. So that you could say that potentially some of these are big browns. The species of interest, little brown, 51% down to 12%. So one of the important things that I see about that is that isn't down to that less than or 99% population decline, at least in Minnesota. In other places they have. Northern long-eared is specifically a focus, and they have always been rare. I had a couple of bat projects before white nose, and the DNR has also done projects, and they're never as common as a little brown. Evening bat is a new species, and the tricolored has also always been relatively rare. Here's the noise and no ID. So basically, in each deployment, we get some portion of noise, which is not a bat, and then we also get call files that are in the frequency of bats, but they can't be differentiated to species. We do get more noise on bridges because of car traffic, proportionately, but we still are recording a lot of bat species. Okay, so that's the effect of white nose. Now let's compare forests and bridges. So this is a 2025 data. So there's 4 times as many calls per night around these bridges. Example deployment here. And, I put like here. That's probably not the best. It's anthropomorphic phrasing, but it works. Big browns are using bridges more than the forest. Silver-haired don't like the bridges, and they are more in the forest. Little browns are using bridges a lot. And then we still got the NLEB and the tricolor being relatively rare. So, it's kind of like one of the things that I've been thinking about is, can we do something that emphasizes the positive aspects of bridges and that they're actually attracted to bats as kind of an offset for any potential take that might occur. Why specific bridges? I still haven't figured this out yet. One possibility, the water versus land that I mentioned. So here's the call files per night on bridges over land, and this could be railroads like here, or roads, or just land, and bridges over water. And my expectation was that there would actually be more action over water because of insects, prey species that are present, but there's more bats on the land, especially the little brown, than over water. So bridge length, we're basically using bridges where that are long enough for bats to use. Surrounding cover types, that would be foraging. Geography, north-south, that is one thing that I will say is that we do pick up on some deployments far more bats in southern Minnesota, District 1, than we do in northern Minnesota. Here's another example, though, season. So the hibernating bats, the cave bats, it's called swarming in late summer, early August, where they get together in larger groups before they go to the hibernacula. And so I was kind of expecting we'd get more call files per night in the late summer and fall, but we actually got more call files per night, especially of the little brown, which is a species of interest, during the maternity roosting period. So that has some implications too. And I didn't point out on the previous one, but this is about 50% deployments. Oh, I didn't update this. Sorry. This is over June, July, and August and October. Okay, a little bit. Let me check the time here. Yep, we're doing okay. The Fish and Wildlife Service uses basically in their protocols, they use what's called a maximum likelihood estimator to determine if a bat species is present or not. And these are calculated by the kaleidoscope and also Sonobat, the programs that we use. There's some indication in the literature in a recent paper that it may not be effective in indicating presence or absence, so that's up for discussion. But assuming that it is, because it's a protocol that is being followed, here we've got the forest, and here we've got bridges, and these are the 3 listed or potentially listed bats. Large number of nights for each of these, and the percent of nights that are present according to this maximum likelihood estimator, less in the forest, but 70% of nights the little brown bat was present. If we look at it slightly differently, this is a percent of deployment. So essentially, how many bridges was the little brown bat present at least one night? And it was present almost all nights, at least one night on a deployment. Northern long-eared and tricolored as well, but those are just, a passing by. Like I showed earlier, they're just never that common in terms of the total number of calls. So now we're getting to the genetics. So I've talked a little bit, is it a day roost or a night roost? The day roost is going to be more important. And there are some bridges which are clearly have lots of bats that are present because there is a lot more fecal pellets. We don't know if with the acoustics, are they just foraging and are they occasionally using the bridges at night? One thing I will say is that we detected bat species, not all 7 species, but in all but 2 deployments. And these 2 could be an equipment malfunction. Basically, they're present throughout Minnesota despite white nose, and the species composition has changed a little bit. So we need to work on the why are they using bridges. And this is what the pellets that we collect, basically, they're picked up with tweezers that are clean, dropped into these tubes, and that's the next step in the process then would be confirmation with DNA. And this is what we're a little bit behind on because of Jessica Sieber's passing. But like I said, we're back up to and running now. But basically, DNA extraction and then PCR amplification, again, done by the grad students and the undergraduates. And then we send them down to the University of Minnesota Genomics Center for sequencing and then processed with this barcode of life database. And I couldn't do this if I tried, but that's what we have younger people for, and that identifies a bat to species. So from the 2024 samples, we got one northern long-eared on a bridge north of Two Harbors and lots of little brown bats. So genetic confirmation of presence. And one of the things about listing species is that if they're listed, take can be defined in various ways, and it doesn't take much. And then Raphael Stern, he hasn't been able to work on the project yet because we don't have enough results for him to do that. We got lots of acoustic results. Genetic work is basically should be done in the next month and a half. We'll have everything down to the genomic center. And what he's going to do is take the DNA results from the bridges and the acoustic results, work his magic with Bayes' theorem, and come up with a probability of presence for bridges that are sampled and bridges that are not sampled, and hopefully extend that to being able to say it's so unlikely that we'll see certain bat species here on these bridge types that we don't need to do any further analysis. One thing, this is next to last slide, most kind of unrelated to this talk, but on another project, we're actually looking at diet composition of bats, and this is from an older student, but we are also doing this on a new project. Bats are mostly eaten Trichoptera, which is stoneflies, mayflies, Diptera is flies, and then Coleoptera is beetles. So that's what, and it's just amazing to me that Miranda picked up almost 600 species, and we've got another 400 or 500 species as well. Oops, what's that? No, I don't know how that happened. Anyway, a couple of acknowledgments are in order. Department of Transportation, MnDOT. Chris Smith laid a lot of the groundwork and came up with the idea for this proposal. Bria Carlson is managing it, and the TAP committee. The forest work was done with ENRTF, or LCCMR as most people call it, funding. And then where the Invasive Terrestrial Plants and Pest Center is what's funding the bat diet work. So we're actually using some of the fecal pellets we collect for multiple purposes. And then lastly, Natural Resources Research Institute helped me and others creating a new DNA lab so we could actually process these results and do it much cheaper than having to send them out. And I will point out, standing there is Colin Burkett. He's been a tremendous help on this project. So with that, I can take any questions because that's the introduction.
Ron, thanks for the presentation. All right. I've been monitoring the Q&A, and so far we have no one brave enough to come forward with the first question.
Okay.
So I'm going to take the opportunity to ask some of mine as people think about what they're curious about.
Sure.
I was really surprised bats can live up to 30 years. Thanks for taking us through all the methods of, detection and observation and genetics. Really like that you tested your land water hypothesis. And fun that you came up with the, no result there or no result. Do we want bats on bridges? And maybe if we do, could we be designing those bridges to enhance the habitat?
Yeah, that is actually being done by some state DOTs. I believe California and another uh, state as well, which I can't remember right now. And that you raise a good question about, do we want bats on bridges? From my perspective, at least, I'm not an engineer, so there may be reasons, but from my perspective, it's suitable habitat. And one thing I like to point out is that, like, for example, um, the Sudan mine, which many of us in Minnesota are familiar with, that's an example of bats taking advantage of a suitable location. And I often think that, given the bats in different mines across the country, that we've actually increased their population.
Hmm.
And especially now, with white nose syndrome, it's a kind of thing where I kind of view it as, if I were Fish and Wildlife Service, I'd view it as a trade-off, in the sense of, yes, we disrupt their lives a little bit when we're doing maintenance on a bridge, or we cut down a tree or two, but overall, the net benefit of bridges to bats is positive.
Nice. Thanks, Ron. Uh, my dad kept bat houses when I was a kid, and his rationale was any enemy of my enemy is my friend, and he had the idea that they're eating mosquitoes. But interesting to see that they're eating all sorts of other stuff. Was mosquitoes part of the diet?
Yeah, actually, we this is fresh in my mind, but Miranda picked them up. Like, I can't remember the details, but there was a study in Wisconsin as well. But Aedes is a mosquito genus, and we have, I don't know how many species of mosquitoes we have in Minnesota, but on the last batch we sent down, we received half a dozen mosquito Aedes um, identifications. So they're definitely eating mosquitoes. It's one of those things a big brown bat wouldn't, because it's so much bigger. They focus on beetles. So it'd
Okay.
be the myotis would be the little brown bat, the northern long-eared, and the um, tricolored bat, which would be able to feed on mosquitoes and actually have a net benefit, especially if they're in a swarm.
Nice. All right, I got three questions that have come in now from Lynn Klarkowski, and I'm going to take them one at a time. All right, so did you say that bridge used by bats is mostly for rest and not for long periods of time? And then maybe the second part of that is the time occupied in roost trees, mostly June and July. So,
Uh,
so wind for both the forests and the bridges.
yeah. So the if you think about it, like, there are definitely bats that are spending an entire day in a bridge. And it's one of those things where we don't, well, we got it's relatively rare that they do, based on our visual observations. Because, like I said, in 6 out of 300 bridges examined, we actually saw the bats physically during the day. So that part is relatively rare. We don't know how many were up hidden underneath the bridges, because they can get places we can't even see, obviously. So the use may be more extensive. What I was referring to on the night roost is that, so basically what they'll do, they'll emerge in the evening at sunset. They'll get their first meal, and then they'll come back. They got to rest somewhere, and they can either rest in a tree, or if they know about a bridge, and based on the fecal pellets we're finding, they definitely are using, like, they would be just, pellets sprinkled about, but not really dense.
Hmm.
So that would be a nighttime thing, and then they'll go out and forage again, and then they'll go back to their day roost. In the tree bat, the bats that are roosting in trees, and this would be the northern long-eareds that we had our project on, for example. Um, I'll say a little bit more about that, but they're doing the same thing. But if there are no bridges around, like the ones in the northeastern Minnesota picture that I showed, they will just find a tree to rest in. They won't go back to their day roost until the end of the night.
Sure.
Red bats, and this is what I was thinking, so more bat biology, but red bats, I've seen pictures of this. I have yet to see it in person, but they will often just roost in trees during the day in leaves. So the leaves on a tree, and if you look up, you'll see a patch of red, and they literally are red bats. That's why they're called red. So,
So maybe to drill down more on Lynn's second question. So when you think about the tree species over the season, is that mostly roosting in June and July, or is it longer than that?
I get you. Okay, sorry, misunderstood that part. Um, they they need to roost somewhere all year long until they hibernate. And so the reason that I focused on June and July is because that's the maternity period when the
Sure.
females are nursing, and it's what Fish and Wildlife Service is mostly interested in with the listing, is the potential harm that would happen, for example, if you cut a tree that had a roosting bat in it.
Right.
Now, given the densities of northern longears, it's highly unlikely in the millions of trees we have out there that they're roosting in that tree, but it's
Right.
possible.
But that's their vulnerable period. So the reason for the
Yeah.
regulation then.
Yep. Yeah.
All right. And then the final question we got right now is what has been learned about eliminating white nose syndrome?
Oh, yeah. Yeah. There's a couple things about that I think, and one of them is that um, the bats that are surviving, and that was one of the interesting things about the calls that I kind of mentioned when I was talking about how we still have a fair number of little brown bats using the bridges. So it's like they haven't gone down completely. So there's a reservoir out there that they are finding naturally probably a slightly warmer hibernacula because the fungus is um, it's there's a certain range of temperature where it does best in. In terms of what can be done, there is some work being done in Pennsylvania that I know of where they're trying to fumigate bats as they come into a hibernacula. Problem with that is that if there's multiple entrances to the hibernacula and that kind of thing, and fumigate to get rid of the fungus is the idea. But how you can push that all the way back into a cave is, we'll see.
Right.
I think the combination of that plus the natural selection for using a different hibernacula will let little brown survive. And with luck, maybe they won't be listed.
Sure. Yeah. So the potential benefit for that selection would be that they would be in a different, maybe less concentrated and in a different temperature
Yeah,
regime that doesn't have as much of the growth of the
yeah,
bacteria or the fungus.
yeah.
Grow-
It's not the concentrate number of bats that are present, but it really is a temperature in the cave
Sure.
that
Okay.
that the fungus is susceptible to.
Yeah. Sounds like nature's already at work on this problem.
Yep.
All right. Well, I was as the engineer here, I was definitely designing something in my head where we have the 12-inch deep and 1-inch wide slots that the bats are really going to love, and then trying to figure out how to form that into concrete. So if you got more ideas on that, I'm ready to start building.
Yeah.
All right. Well, we don't have any other questions coming in, so um, we can um, bring Maurizio back for our closing or transition.
Thank you, Russ. And we can see if we can do a quick round robin if anyone is interested in sharing, um, what transportation issues are you currently working on?
Sure. Yeah, I guess so. As we close out the speaker, the presentation, I guess thank you, Ron, for coming in and for giving us this tour of your work. We're really excited to see it finalize and read the final reports. Hopefully, this is valuable for you as well when you think about some of the questions on methods and the life of bats. You definitely sparked our interest.
Yeah,
Thank you very much,
things
Ron.
like
Yeah.
this are always beneficial. Makes me think.
All right. So the last part of our meeting today is about round robin. We like to open it up for folks if they have any specific transportation related issues related to energy and the environment that your organization is facing. We're always hoping to get some input to help us in our mission to connect university researchers, agency partners, and practitioners. So feel free to use that chat to put in anything, or you can always raise your hand to request to have us unmute you. Maurizio, anything your organization or any ideas that Ron sparked today for you that you can see connecting on?
I cannot think of anything, but it was really interesting to learn about bats. I know that they're really important for the ecosystem. So it's just good to hear that our infrastructure can serve that purpose of also providing habitat without harming the infrastructure. Of course, that seems like a really great thing to know that that type of research is available.
Yeah, this topic got me really thinking about, are there other ways that I know we're always thinking about like fish and creek crossing for culverts, but there's all sorts of connections to habitat and the built environment. We're intentionally putting green roofs on, and our stormwater features are always trying to serve those multiple benefits for all sorts of habitat. Um, had never thought about a bridge. I'm excited to kind of take that same sort of habitat thinking more in a vertical way. So um, I'll definitely take this new idea into my work, and maybe it has some connections to others in this area where they can start to see it in a little bit different way. All right, looking at the chat, I'm not seeing anyone else with specifics on what their organization um, uh, issues they're facing with environment. We'll just put out a last call for that. The other questions we have are, what ideas do you have for potential future council meetings or topics? And then, are there any other organizational or informational events that we can think that this audience might find valuable? For that one, there's a national conference coming to Minnesota this year. StormCon is coming. So for those in the water resource fields, we've got a national conference coming. Take a look for that. We mentioned at the beginning the other CTS events. So take a look at the that CTS webinar or the CTS links for some of those future webinars and the in-person events. Anything else coming to mind for you, Maurizio?
Not really,
All right.
but those are really good ones. Thank you for bringing them up. I think that if there are no additional comments, I think that maybe we could give you some time back. Uh, Thank you so much again, Ron, and thank you, Ross. Thank you, Sam and James. This has been a great webinar, and we really appreciate all your participation. I hope you can join us for the next one, and thank you.
Thanks again, Ron.